Hello. My name is Theron, and I am here to tell you about Golgi bodies or Golgi body, the post office of the cell. The Golgi apparatus, also known as the Golgi complex or Golgi body, is an organelle found in most eukaryotic cells. It was identified in 1898 by the Italian physician Camillo Golgi and named after him. Part of the cellular endomembrane system, the Golgi apparatus packages proteins inside the cell before they are sent to their destination. It is particularly important in the processing of proteins for secretion. Structure. Found within the cytoplasm of both plant and animal cells, the Golgi is composed of stacks of membrane-bound structures known as cisterna, singular, cisterna. Each cisterna comprises a flat, membrane-enclosed disc that includes special Golgi enzymes which modify or help to modify cargo proteins that travel through it. Each cisterna comprises a flat, membrane-enclosed disc that includes special Golgi enzymes which modify or help to modify cargo proteins that travel through it. The cisterna stack has four functional regions: the kissed Golgi network, medial Golgi, endos Golgi, and trans Golgi network. Vesicles from the plasmic reticulum, via the vesicular-tubular clusters, fuse with the network and subsequently progress through the stack to the trans Golgi network, where they are packaged and sent to their destination. Each region contains different enzymes which selectively modify the contents depending on where they reside. The cisterna also carry structural proteins important for their maintenance as flattened membranes which stack upon each other. Here are some functions performed by Golgi bodies. Cells synthesize a large number of different macromolecules. The Golgi apparatus is integral in modifying, sorting, and packaging these macromolecules for cell secretion, exocytosis, or use within the cell. It primarily modifies proteins delivered from the roughened plasmic reticulum, but is also involved in the transport of lipids around the cell, and the creation of lysosomes. In this respect, it can be thought of as similar to a post office. It packages and labels items which it then sends to different parts of the cell. Enzymes within the cisterna are able to modify the proteins by addition of carbohydrates, glycosylation, 12 phosphates, phosphorylation. In order to do so, the Golgi imports substances such as nucleotide sugars from the cytosol. These modifications may also form a signal sequence which determines the final destination of the protein. For example, the Golgi apparatus adds a -6 phosphate label to proteins destined for lysosomes. The Golgi plays an important role in the synthesis of proteoglycans, which are molecules present in the extracellular matrix of animals. It is also a major site of carbohydrate synthesis. This includes the production of glycosaminoglycans, GAGs, long unbranched polysaccharides which the Golgi then attaches to a protein synthesized in the plasmic reticulum to form proteoglycans. Enzymes in the Golgi polymerize several of these GAGs via a xylose link onto the core protein. Another task of the Golgi involves the sulfating of certain molecules passing through its lumen bisulfitrontraces that gain their sulfur molecule from a donor called PEPs. This process occurs on the GAGs of proteoglycans as well as on the core protein. The level of sulfatin is very important to the proteoglycan's signaling abilities as well as giving the proteoglycan its overall negative charge. The phosphorylation of molecules requires that EPP is imported into the lumen of the Golgi and utilized by resident kinases such as casein kinase 1 and casein kinase 2. One molecule that is phosphorylated in the Golgi is apolipoprotein, which forms a molecule known as the LDL that is a constituent of blood serum. It is thought that the phosphorylation of these molecules is important to help aid in their sorting for secretion into the blood serum. The vesicles that leave the roughened plasmic reticulum are transported to the kiss face of the Golgi apparatus, where they fuse with the Golgi membrane and empty their contents into the lumen. Once inside the lumen, the molecules are modified, then sorted for transport to their next destinations. The Golgi apparatus tends to be larger and more numerous in cells that synthesize and secrete large amounts of substances. For example, the plasma B cells and the antibody-spreading cells of the immune system have prominent Golgi complexes. Those proteins destined for areas of the cell other than either the plasmic reticulum or Golgi apparatus are moved towards the transface, to a complex network of membranes and associated vesicles known as the trans Golgi network (TGN). This area of the Golgi is the point at which proteins are sorted and shipped to their intended destinations by their placement into one of at least 3 different types of vesicles, depending upon the molecular marker they carry. The transport mechanism which proteins use to progress through the Golgi apparatus is not yet clear. However, a number of hypotheses currently exist. Until recently, the vesicular transport mechanism was favored, but now more evidence is coming to light to support cisternal maturation. The 2 proposed modules may actually work in conjunction with each other, rather than being mutually exclusive. This is sometimes referred to as the combined model. Cisternal maturation model. The cisterna of the Golgi apparatus moved by being built at the kiss face and destroyed at the trans face. Vesicles from the endoplasmic reticulum fuse with each other to form a cisterna at the kiss face. Consequently, the cisterna would appear to move through the Golgi stack when a new cisterna is formed at the kiss face. This model is supported by the fact that structures larger than the transport vesicles, such as collagen rods, were observed microscopically to progress through the Golgi apparatus. This was initially a popular hypothesis, but lost favor in the 1980s. Recently it has made a comeback, as laboratories at the University of Chicago and the University of Tokyo have been able to use new technology to directly observe Golgi compartments maturing. Additional evidence comes from the fact that Kebbi vesicles move in a retrograde direction, transporting the plasmic reticulum proteins back to where they belong by recognizing a signal peptide. Vesicular transport model. Vesicular transport views the Golgi as a very stable organelle, divided into compartments in the kiss to trans direction. Membrane-bound carriers transport material between the plasmic reticulum and the different compartments of the Golgi. Experimental evidence includes the abundance of small vesicles, known technically as shuttle vesicles, in proximity to the Golgi apparatus. To direct the vesicles, actin filaments connect packaging proteins to the membrane to ensure that they fuse with the correct compartment.